Hi, everyone. It's great to be here, and I would like to represent Innotech Precision Medicine. My name is Roya Khosravi-Far, and we are working on the next generation of diagnostics for multi-omics detection. So I have a snapshot of Innotech Precision Medicine. We're headquartered in Boston, Massachusetts. We're founded by a group of life sciences and engineers from Harvard Medical School and Massachusetts Institute of Technology. We are funded by RADx for development of radical diagnostic technologies. Additionally, we have had funds from NIH and other non-dilutive fundings and some angel funding. We have a strong team working with engineering and contract research organizations for time and cost-effective manner. And our mission is to engineer the next generation of multi-omics diagnostics. Our program will be ready for FDA and CE submission with about $6 million in additional funding. So Innotech was founded based on our belief that early detection saves lives by enabling timely and effective treatment and even, in some cases, prevention. For example, in the last decade, we've had significant advances in cancer therapeutics. However, despite these advances, in 2019, there were more than 23 million new cases of cancer that were documented globally, and more than 10 million people succumbed to cancer. So even the best therapeutics cannot work if these kind of diseases, these complex diseases, are not detected early enough. Most of the initial diagnosis and screening for many of the cancers is based on in vitro diagnostics. But the current in vitro diagnostics have high rate of false results, and they are not good enough. The gold standard of in vitro diagnostics is still nucleic acid detection. However, nucleic acid detection it shows predisposition to the disease, but not the actual disease. It's the protein expression that is actually providing the evidence for the state of the disease. If we can integrate nucleic acid and protein tests in the same test, having the same device with the same statistical analysis and readout, then we can have a better, more accurate diagnostic strategy. At Innotech, our patented technology is the first in class for multi-omics detection. We have developed our chemistry and our disease-specific cartridge for development of multi-omics detection by multiplexing detection of currently nucleic acids and proteins. In the future, we're going to add epigenomics and metabolomics as well. And our device is going to control the functions of our microfluidic, and also it's an ultra-portable device that can be used at the site of the patient with ease of use and for providing rapid results. Our technology provides high-performance multiplexing as well. So our chemistry and our microfluidic is a plug-and-play technology that basically, depending on the disease biomarkers, we can generate very quickly chemistry and cartridges for different indications. Our nucleic acid detection is based on LAMP combined with CRISPR in one pod. LAMP assays provide a rapid PCR test. However, LAMP assays have a higher rate of false positives because of the primer binding. CRISPR provides an exquisite selectivity to our nucleic acid detection. Our protein detection is based on a modified ELISA assay that is detected by fluorescence and signal amplification. So we start from the same specimen and process that specimen within our all-enclosed microfluidic cartridge for detection of both proteins and nucleic acids. So our initial application is in women's health, and our initial indication is in cervical cancer. Cervical cancer cancer, according to the World Health Organization, has been identified as a major global health concern. And the global market for this by 2030 is going to be $38.2 million. We anticipate to go for US regulatory approval by 2026, and we already have had initial discussions with FDA regarding our path as a de novo 510(k). So cervical cancer is one of the most common cancers in women. It is curable if it's detected early, but unfortunately, more than 50% of women who get pancreatic cancer die from this disease. And the main reason is because the disease is not detected early enough in these patients. More than 90% of cervical cancers are caused by high-risk HPVs, which is the most common sexually transmitted disease. However, HPV detection alone is not sufficient to demonstrate cervical cancer. More than 80% of us, will have HPV infection at some point in our life. But fortunately for us, our immune system will get rid of that HPV infection. So the presence of HPV doesn't necessarily mean that we have cancer. However, when the high-risk HPVs integrates into the cervix and they result in activation of protein progression markers, then they initiate a cascade of events by changing activation of oncogenes, inactivation of tumor suppressor genes that result in progression of cervical cancer. So at Innotech, we have been able to develop our chemistry for rapid and accurate detection of viruses, including HPV. We can detect down to about 100 viruses in real time in less than 30 minutes with our combination of LAMP CRISPR assay. We can detect our proteins at low picomole levels, and in patient specimens, we have been able to discriminate between healthy and normal specimen in concordance with clinical data. So our test provides an accurate, fast, and portable and easy-to-use device. So of course, there are competitions in cervical cancer, but our differentiating factor with our competitors is that we are the only company that can do multi-omics detection of both nucleic acid for HPV and the cancer protein markers simultaneously. And Quest Diagnostics has performed studies on 13 million specimens for over 10 years period, and they found that HPV testing alone has more than 40% false positive and false negative results, and also cytology alone also has more than 45% false result. So combining the proteins and nucleic acids in the same test with the same process will provide us more accurate results. We also are the only company that actually have a portable point of need device, while the other devices are actually a bench top, and our cost is less than $5,000. So our business model is a razor blade business model, and basically through the sale of, if by year 4 we have 10,000 units that is used 8 times in 300 days, we can generate close to $400 million. So in the last 2 years, with the funds from RADx-RAD, we have developed our chemistry, we have our initial prototype for our microfluidic, and we have a breadboard device. In the next 2 years, we're going with additional $5 million that we're raising, we're going to finalize our design-locked system, do analytical validation, and manufacture a beta system for our clinical study and submission to FDA. We have a strong team of leaders that are experienced in molecular diagnostics, translational medicine, engineering, product development, and business development. And we're working with advisors and partners at MIT, Harvard Medical School, as well as Radix, and we're working with engineering firms. Thank you very much for your attention, and I will be glad to take any questions later. You can connect with me, and we can explain more about our program. Thank you so much.